Okay. My name is Chase Graham,
And I'm Ellie Mizue.
and our project is using machine learning to predict traits from genetics of Oryza sativa or rice organisms. So we'd like to give a thank you to John Kay, which made all this possible and gave us the opportunity to conduct research for our project over the summer.
So we're going to start off with a little bit of basic information or background to help understand what our project aims to do and some fundamental ideas. So DNA is the building block of life, which encodes for information about an organism. For a quick review on what DNA is, it's made up of nucleotides that each have a phosphate group, a sugar, and a nitrogenous base, which is either adenine, thymine, cytosine, or guanine. These nucleotides then pair up to form DNA, A with T and C with G, in long strands. Two sets of these long strands then pair up to form double helixes, the figure we can commonly recognize as DNA. These long strands contain information that translates to the creation of proteins, which play a huge role in the functioning of our bodies, as well as all organisms. Just as DNA is made up of nucleotides, proteins are made up of amino acids. In our cells, first DNA is transcribed into RNA, and then that RNA encodes for a polypeptide, which forms a protein. Every 3 RNA nucleotides encodes for 1 amino acid and is called a codon. Through these processes, we can understand that the proteins that make up the color of our eyes or our hair are all based on the DNA in our cells. And so all of our features are a result of our DNA. Single nucleotide polymorphisms, or SNPs, are variations in DNA that can result in altered characteristics or traits. For example, this image shows an example of a SNP, where the general population would have an adenine, where those who have that particular SNP would have a guanine instead. SNPs occur all throughout a organism's genome. For example, in this image, which displays a map of SNPs across all of an organism's chromosomes. The simplicity of SNPs, as well as their prevalence, make it a good way for scientists to analyze variation among genetic material, as well as the impact of variation on features. And these are the same reasons we chose to use SNPs to analyze the rice genome. In our project, we use neural networks to help analyze SNP data. Neural networks are statistical models that identify patterns through an input and an output through repeated trial and error. They contain many different layers that are inspired by a human's brain structure and function, which is displayed in this image. Through various trial and error, as well as huge inputs of data, neural networks are able to adjust their weights of certain values in each of these layers to accurately make predictions or classifications. Moving on to our purpose, our main purpose was to use machine learning technology to help predict phenotype or physical features displayed from genotype, a species genetic material, in rice in order to better identify the relationship between genotypes and phenotypes. Current methods involve biologists creating statistical models in order to identify which genetic mutations cause changes in a population's characteristics. These statistical models can be very complex and tedious. For example, the image below displays a genome-wide association study, or GWAS, in which the correlation between SNP and the trait is graphed for thousands of SNPs of SNPs along the genomes, in order to determine which areas in a chromosome have the most effect on a trait. Then scientists use high correlation SNPs or locations of interest and later use that data to try and determine which variations of organisms of rice are ideal for creating strains of rice that are most efficient. Although it can be difficult using common statistical models, a machine learning model creates its own weightings or formulas to predict phenotypes. Because of the complexity of machine learning, neural networks are able to detect and predict more complex patterns and correlations, which would usually be tedious to do. Our project aims to increase accuracy in genomic selection for crop breeding programs through detection of more complex patterns. We aim to harness machine learning's ability to quickly and accurately detect these patterns in order to predict phenotypes of rice based simply on a sequence of their SNPs. This would ideally help to better understand the relationship between genotype and phenotype, as well as help to find more robust strains of rice.
Okay, so now we're going to move on to our methods. And so the first thing that we did is we compiled a data set from public sources that contains the SNP sequences and trait data for thousands of rice organisms. So we had 2,266 samples with 150,000 SNPs each. And we filtered this initial set of SNP strands using a process called linkage disequilibrium and minimum R squared filtering. And then each organism had 12 agronomic traits, including heading date, grain weight, culm diameter, and other plant traits. And then on the right, you can see a sample of our data set. Each organism has the SNP sequence and then the 12 agronomic traits. So then we had to encode this data set in a way that a mathematical model could understand. So since each SNP right now is just a solid strand, we had to break these up and each of the qualitative trait data into basically what you can think of as 150,000 dimensional vectors, which we then pre-processed and plugged into our models. So we made three models in order to compare, and we made a dense neural network, a convolutional neural network, and a linear regressor. So right here, you can see the diagram for a fully connected feed-forward neural network, also known as a dense neural network. So basically, first thing happens is that your input data goes through a dense layer, which is basically just, it connects all the neurons from this layer to the previous layer and the next layer. And then it goes through a bunch of compilation stages called batch normalization and neural dropout, which is basically where you just cut off a certain portion of the neurons. And then I'll iterate through the cycle a couple of times before going through a final compiled dense layer and then going into output. So basically what happens is we give it 2,266 samples with sample inputs being SNP or SNP sequences and the outputs being traits. And then the data will pass through each layer where it goes through this process of dense normalization and then neural dropouts. And at which point the model will begin to make associations and weightings in this process, which we call training. And then in the end, we'll give it some SNP sequences and it will be able to predict the traits that the sample that has that SNP, that SNP sequence will have based on its internalized parameters or weightings. And then the feed-forward neural network is a very basic kind of neural network. It excels at basic pattern matching, but it kind of struggles with complex data sets or sets with very specific needs. So data typically needs to be heavily filtered or pre-processed in order to be effective. So this next model is a convolutional neural network. During training, what happens is the data is fed into the model as SNP sequences alongside the traits known as sequences, same as in the feed-forward neural network. And then data first passes through a convolutional layer, which uses this mathematical operation called a convolution in order to assign the weightings. And then it goes through a series of composition layers called max spooling and flattening before returning to the initial convolution layer. It goes through a couple cycles of reiteration before going through a final dense layer and then into the output. So after training, we'll feed it a SNP sequence and ask it to tell us what traits an organism with that sequence will have. And then some of the data is originally put into training, but a small portion of that data will be put into testing. So we can give it some samples it hasn't seen yet, and it'll be able to tell us what it thinks those samples will have, and then we'll be able to gauge accuracy based on that. And then the convolutional neural network, it works great for complex data sets because it's really good at catching tiny nuances in large, complex amounts of data. Like these types of models are usually used for image processing, where models will have to sift through what can be like millions of pixels usually, and then be able to make associations between a ton of inputs and a ton of outputs. But it does require a lot of data in order to be accurate. And then our final type of model is called a linear regressor, which is a very simple type of machine learning model, but it is not a neural network. So basically what it does is it fits all the data to a line, like in the image you can see right here, and then you ask it to predict something that isn't in the original training data. What it can do is it can extrapolate the existing data, or like you give it sample inputs and then it'll extrapolate or it'll interpolate between the two closest values, basically. And then it's the least complex data set, has no recurrence at all, which is just feeding the data back into the model once it's trained. So it's really easy to set up, and it gives pretty reliable results, but it'll definitely struggle to capture some of the complexities when the data is really large. So yeah, what happens is we give it a bunch of SNP sequences, and for each SNP, it'll look at the association between changes in that SNP in the data and then changes in each trait in the data. And then using the linear regression and prediction process, it'll be able to tell us things about what associated traits value will be. And then so we chose these three models because they cover a wide range of machine learning models. It's useful to compare them, and then we can get a lot of insights of how the complexity of the model fares against the complexity of the data set. And our project was actually just originally for convolutional neural networks, but we wanted to add other models so that we can compare. And some of our models were actually trained on all of the 12 traits at once. We call these multi-target models. And then some traits were trained on all 12 traits individually. We call these single target models in order to give us properties into the other insights, or insights into the other properties of our models.
So now that we have a better understanding of the process we went through, we can go through some of our results. We measure the accuracy of our models using something called a Pearson correlation coefficient, and this measures the correlation between each of our model's predicted values and then the actual values associated with the actual organism's traits. In a Pearson correlation coefficient, it's given on a scale from 0 to 1, in which values closer to 1 are more accurate and values closer to 0 are less accurate. This chart displays all the different traits our models tested and attempted to predict, as well as the Pearson scores for each of the traits and for each of the 4 models for each of those traits. Highlighted in red are the highest scoring model and trait combinations for each trait, and highlighted in green are the highest performing model for each of the traits. For example, if we look at leaf length, the multi-target dense model performed the best among all 4 models, and the multi-target dense and leaf length combination was a high scoring Pearson score overall. This chart displays another way we can visualize our data, again comparing the results for all 4 models across each trait. In this graph, we can clearly see that some traits performed better than others, and our machine model was better to accurately predict the phenotype for certain traits than others. For example, across all 4 models, heading date and comb length were more accurately predicted compared to traits like comb number or seedling height. As shown in the previous bar graph, model performance varies for certain traits. This can be attributed to the fact that certain traits may be more heavily dependent on the environment rather than genetics, which can make it difficult for neural networks to accurately predict the trait based solely on SNPs. These graphs display the predicted values on the x-axis and actual values on the y-axis. Ideally, the values would line up perfectly for a slope of 1. However, as we can see for these 2 traits, heading date and comb length, the values have a high correlation, which corresponded to higher Pearson scores across not just one of the models, but all 4 models. And then this diagram can help you to better understand what these traits mean. So the heading date is the date at which 50% of the florets have bloomed, and then the comb length is the length of the stem or the comb of the plant.
So model performance also varies for certain traits because traits depend differently on an organism's genetics and the environment. So as you can see in here, the data it's not very linearly distributed. It's kind of random and all over the place, which shows that there's less correlation between the prediction values and the actual values. The reported score for each of these graphs was probably a lot less than those on the other traits. So some traits had excellent performance across models. Some had great performance only on certain models, and then some traits did not perform well across all models. And all this is due to varying individual model performance and also varying heredity across traits. So right here, you can see that this the seedling height is the trait we were talking about in the last one. It's basically just the height of the individual plants. And then across multi-target and single target models, performance also varies. Right here, you can see that for grain width and grain weight, single target dense performs significantly better. But for leaf length, multi-target dense performs a lot better. So what we can get from this is that this is just the nature of having a model try and associate in really complex data with a ton of variability, which would be multi-target, or you could also choose to sacrifice the access to other parts of the data set, which might be necessary to really solidify these associations. So it's kind of a trade-off, and we just had to select which models work best for which traits individually. And then right here, you can see leaf length, that's the length of the leaves right there, and grain width, just width of the grain. So we performed a statistical test called sensitivity analysis to determine how trained test split ratios affect the model's accuracy. So on the right side, you see that more of the data, a greater proportion of the data was put into training than it was for testing. So on the right side, we were really uncertain about how the reported Pearson scores are. But on the left side, there was a lot less testing and a lot more training. So we're really certain about those, but as you can see, the accuracy was worse for multi-target dense and single target dense. But for a multi-target convolutional, we can see that its performance stayed very still, which indicates that we probably need a lot more data. So convolutional neural networks typically rely on a large amounts of data in order to make associations. So a CNN was not the most appropriate choice of model for this task. As you can see in the plot, the validation accuracy, that's just the accuracy for testing, stayed pretty solid across training times. And then, yeah, so our results compare favorably to results of previous studies. You can see on the diagram on the right, that is the results from previous study. And on the left, those are the results of our study. So yeah, you can definitely see that certain traits have higher heritabilities. It's reflected in both tables. And then some traits are higher than others, like heading dates. And yeah. So our technology has applications in crop breeding programs, human disease research, and medicine development. So in crop breeding programs, basically what you just have is you have models that look at a sample of crops and sequence for DNA in order to optimize these breeding programs and get certain traits, select the traits that you want. For human disease research, you'd use the same technology, applying it to humans to identify early signs of having hereditary diseases, and you can get early treatments. Currently, what happens is that researchers will sample a whole person's genome and run a GWAS on the entire population to try and identify these threats. And also in medicine development, you use the technology to develop medicine and other forms of disease treatment. So over the summer, we learned a lot about machine learning models and their applications in genetics. We also gained valuable scientific research knowledge by figuring out how we're going to approach this problem and creating procedures, writing reports, and we also, of course, strengthened our teamwork skills.
Thank you.